Thank you. Thanks for the invitation. There might have been a small mistake because I'm an experimentalist.
Don't call yourself a mistake.
Oh, it's going to get worse. But I thought, since you invited me to a spin glass meeting, I would do my best impression of a theorist. But the problem is
that why you're taking off your shirt?
No, I'm buttoning this.
Your shirt.
No. So I'll do my best impression, which is to say that I'll present something that I don't fully understand, and I'm relatively sure of, and it will end with a bold claim, which may or may not be true, but I think it probably is.
The definition of theory.
Yeah. Yeah. So what I want to talk about is how we should think about interactions in microbiomes. And I've been working on microbiomes for a long time. Actually, when I was a PhD student, I did low temperature spin physics, but those days are long over, so the Hamiltonians are all gone. And I've been thinking about these systems lately, which are the microbiomes in soil particles and so on. And I should also add, in the spirit of a theory talk, this was very hastily prepared. So slides don't work. It's my fault. How do we think about interactions in these systems? So if we run a sequencer on some soil, we see, hundreds, thousands of species. And I think our assumption is that the interactions in those systems are really complicated. And hence, maybe we should think about them as a spin glass. Because there's a random energy landscape and so on. My message is that we should not think about these systems as spin glasses. That in fact, the underlying structure and architecture in them is much simpler than that. And this will be my argument today. So interactions are complex, right? I want to talk about interactions in a specific context, which is motivated by the biological problem that we're working on, which is in the context of what we term function in these microbial communities. So microbial communities in the lake, in the soil, and so on, they perform metabolic processes, they perform biotechnological processes that have some functional sort of emergent output, okay? That would be like the rate of some chemical reaction. Doesn't matter what it is. I'm sorry, there's actually chemistry there. I should have deleted it, okay? It could be
carbon, so it's inorganic.
Yeah. Anyway, the way we think about interactions is, imagine you have some community with some set of interactions, got some function, and the problem I want to address is what happens when I delete a member of the community. I want to know how that affects the function of the system. And in principle, if this is a spin glass-like system, that's a very hard problem because I have a lot of interactions and I've deleted some component of the system. And now I went to some other crazy place on this rugged energy landscape, for lack of a better term, and I'm lost, right? This prediction problem should be very hard. Okay. So the function is something we sort of need to hold on to have a thing to predict about these systems, which is why I'm thinking about it. One way to do this, and people have done this, is to say, okay, I'm gonna make a dynamical systems model of this ecosystem. Okay, so I'm gonna parameterize these arrows. I've done it myself in the lab. It's very painful, and I'm gonna get a model for the dynamics of the system, and then once those dynamics settle down to some fixed point, let's say, I'll predict the function somehow. And then if I have that dynamic model and I delete one of those species, I get some other dynamics predicted by my knowledge of the interactions in the system, and then I get some other function, I'm done. Okay, but that's a hard way to go because I've really parameterized the interactions in the system. And the presumption going in is that those interactions are really complicated. So one of the approaches to not do that, which is very popular because of you spin glass folks, which was popularized in genetics really a long time ago, is to forget about all that nonsense and let's take a statistical approach. Let's just think about landscapes, okay? In genetics, this is very popular and I suppose in many ways useful, where one has a genotype space, which is itself, of course, a high-dimensional 2 to the n object. And that's got fitness peaks and valleys on it and so on. And as I move around in genotype space, there's experts in the room on this, I go up and down this fitness landscape. In the community problem, I can define an analogous landscape, which isn't mutations, but instead the composition of a community, presence, absence of strains. And there are a couple of papers that were on the last slide where we started doing this quite seriously, actually, with several collaborators, including Mikhail, who thankfully is here and can bail me out when the theory doesn't work out. And so I'm going to try to do this on a data set right now. So here's the data set, not acquired by my lab, actually. This is from Ophelia Venturelli at Duke. So what she does is she's got bacteria from the gut, 25 of them, and they produce a compound called butyrate. Again, doesn't matter, but she cares about that. Human gut people care about that. And what they did was a really horrifying experiment, very expensive and slow and hard, where they took 500 combinations of these 25 strains, and they just put them in a dish and they grow them for some fixed period of time, and then they measure the butyrate. That is the function. It's a scalar quantity. Okay? And that gives them a data set, which is something like this, right? They vary the community composition. So they're moving around on this community function landscape. They vary the community composition. So for each row of this matrix, which I'm encoding as plus or minus 1, which I'll talk about in a moment, they have some measurement of the function of the community butyrate. It's a concentration, okay? And so now what you can do is, there's a very nice paper from Ramaranganathan and Frank Polvik 10 years ago or so, delineating that if you encode presence absence in plus and minus 1, this is a Fourier basis when one does a regression of the following form. So imagine I want to predict the function of my community. I'm doing a Fourier expansion on this landscape. That's all that's happening. And the Fourier expansion, has a zero mode, essentially, additive terms, epistatic terms, and so on. I can go up to whatever order I want. And, with 500 data points, I can only fit so many of these coefficients. But the question I'm going to ask is how many of these interactions, statistical interaction, I think one should not interpret these as ecological interactions per se, but we'll get there. We'll get to that. How rugged is this landscape?
So can you clarify the function is the energy itself creates? I missed in this picture how is the function and the energy landscape related, and what happens if
the function is the energy landscape, right? I'm only talking about scalar functions right now.
I see. So this assumes that there's one.
There's one, and I choose what it is, and it's arbitrary.
There's several, and so all of these things will depend on which choice you make.
It will. Yes, absolutely. Yes, absolutely. That's true.
And somehow, strangely enough, also the definition of grouping of species may depend on that.
Absolutely.
It's going to screw up.
Absolutely. This landscape is a fragile object in this problem, right? And we're going to get to the fragility, I think. Okay.
So you said that this like a Fourier basis, but you instead of the x's, you could have written in Fourier space and clumping them together in different ways.
Yeah. So basically, the way to think about it is, these coefficients, when the independent variables of this regression are plus and minus 1, are Fourier amplitudes on this landscape. The details are in this paper. It's a
Fourier basis of the hybrid.
Yeah. That's it.
I have another question. So it's only presence or absence, but of course, in any given community, you'll have different frequencies of these species.
Yeah.
So that's sort of completely.
Yeah, all gone, out of here. Yep. Yeah, same is true for genes, right? If I have a mutation, that could just affect the abundance of the transcripts. But I buried that in the presence and absence of a mutation. I think it's totally analogous. Okay, so it turns out that I can predict a lot of the function with additive effects. And I do a little bit better if I add second order coefficients. And I do a little bit better if I add third order coefficients. These are in and out of sample. Okay, so it looks like the landscape is mostly dominated by these additive terms. And if I take a quadratic model, that means up to this order, and I plot my observed function versus my predicted function, not bad.
But if I take up to third order, say, and do an L1 suppression to try to make it sparse.
This is already L1 suppressed because we're in big trouble in this model, right? Because there's 25 species, which means 25 squared over 2 of these second orders. We can't infer third order reliably here, but I'm going to get around that.
But you're saying even after you do suppression, it tends to be dominated by the lower orders.
Yes. But one could worry that a regularization was artificially suppressing epistasis that was actually there. You just couldn't see it and it buried it in low order terms. The answer to that is we have a data set that's combinatorially complete. We measured every combination and we still see the same pattern. Okay, so I think there's a real property of these landscapes. That's what I'm going to say. Okay, so this is, when you're in this Fourier basis on the hypercube, basically the fraction of the variance explained from each order of the model can be just read off. Okay, that's great. And the punchline of this paper from Abby Squara, who was sort of an orphan at Yale when Alvaro Sanchez moved to Spain and just started hanging out with Mikhail and I. We wrote this paper in a period of 4 months, maybe. Basically, she did this on all of these data sets, and the story that I just told you holds. Yeah. And we were very excited. Actually, we've even done engineering with this approach. So we take libraries of strains. We have a paper that came out last year in Nature Microbiology and shows that we can design synthetic communities by training this model, using the model to predict the function, and then we actually inoculate them in the soil and they perform the function that we designed them to do. Okay, so in some sense, you should take the model seriously.
Even though your laboratory conditions are extremely different from the soil conditions.
Yes, but there's an important reason that worked, which is that they were targeting a niche that was open in the soil that we inoculated them to. Let's talk about it later.
Naive clarification: this object you constructed as like an energy is not a generator of a dynamics, though.
It only predicts an endpoint static quantity. Yeah.
There's no sense that you go up that landscape.
No. I mean, these are the dynamics that I explicitly got rid of, although they will come back at the end. Yeah. Okay. So when we wrote this paper, Mikhail and I, he was doing a sabbatical in my group, super fun. He was like, should I even be surprised by this? Which I think is a very smart question. And the short answer is no. Okay. And how do you get the short answer? Here's a numerical answer to should I be surprised? So what Mikhail did, and maybe if I fall off a cliff here, he can bail me out. But one generates a bunch of LV models, Locke-Volterra models, with 10 species. And then there's this phase diagram from Guy Boonin and others, where one varies the average interaction strength and the variance of interaction strengths that one draws these interactions from a random distribution. And what we're plotting here, what Mikhail is plotting here, is the function is now the total biomass in the system, like the abundance of all the organisms summed up. And what he's plotting is the R squared of a second order model on presence absence to predict that function. And basically it works everywhere. That's saying if you have a Locke-Volterra model generating your dynamics, this landscape picture that we're using should just work. And then
Volterra model also second order?
Yes. Yes, correct. Okay, so we really wanted to break it somehow. So Mikhail successfully broke it by building essentially trophic chains. So these are like compound 1 goes to 2, goes to 3, goes to 4, goes to 5, goes to 6. That's this axis. And the fraction of variance explained by first order, second order, third order coefficients in the model, basically your first order coefficients stop working when the trophic chain is really long. Okay, it's not so important for my argument. The point is, when you have a consumer resource model where you have organisms consuming a resource and spitting out another resource, you're in this regime, and your regression coefficients in the additive in that first order term, they work quite well. They explain a lot of the variance. Okay, and if you make the system ecologically much more complex, meaning it has this trophic chain, then you can break it. But I'm not going to push on that further. The point is, don't be surprised. Okay. So I want to make one other point before I go on to the next. Yeah, that's this point. Basically, for all of these models and these models, I only need n squared numbers to specify this landscape. The first and second order coefficients are pretty much enough. I got it. Okay.
What is n again?
Sorry, n is the number of species. Thank you. This is the sloppiness of the talk. It will probably be something else in a few slides. Okay. Yes, they're symmetric. Yeah.
It does.
Sorry, Vincenzo. I know it's.
Not that you're a boring person.
There's none of this. I don't get to color my fingers.
But then it means that you also have an actual energy in addition to your, right?
So your logical terra model isn't symmetric. Not with cost feeding.
The lockable Terra model is symmetric now.
Cost feeding?
So the second model here was a consumer resource model, not a lockable Terra model. I'm sorry, I don't think I said that very clearly. This is a lockable Terra model.
I don't. But they are symmetric or not? Probably symmetric.
I think symmetric, according to the guy who wrote the code.
So that means that there is actually an energy that.
But the resource
Right.
model is perfect.
Is basically I have an organism that consumes one thing and spits out another thing,
But the question
and that
whether it
defines my
does
function.
Perfect efficiency.
Perfect efficiency? No,
Also, whether it turns back into something symmetric. I mean,
that I don't know the answer to.
the usual way people write down consumer resources model is also a Lyapunov function.
That's correct, I believe.
It can be interesting to compare that function, that is a generator of the generator of dynamics,
Yeah.
to the other objects that you could. I don't know if there's an obvious way to understand.
I think I'll get to some very pedestrian, bad theorist version of that at the end.
For this, you're showing actual like experimental data.
Yeah.
And at this like results, does
Yeah.
it mean that those experimental data, like, at least those like community does not have like this multi-tropic level?
It doesn't, yeah. And in fact, we know that biologically in those systems. It's an astute question, yes. I'm going to switch gears. There's another way to look at these landscapes, which is popular in genetics and was popularized by Alvaro in communities as well, which is to study patterns which have been called for the last 10 or 20 years global epistasis. Okay, so imagine I have some community, these three species, and they have some function F, whatever it is. That's like the butyrate that was produced, the biomass that was grown, whatever. And then I perturb that community by adding a target species, the orange, and I measure an F prime. So that target species perturbed the function of the system in some way. It turns out that one can plot delta F, which is F prime minus F, as a function of the background F that I perturbed. And one gets a straight line. One calls that global epistasis. It's a definition by construction. Okay? The bad theory is coming soon. It's where I relate these two. Okay, it turns out Alvaro published a paper last year. I'm sorry, there's a bunch of authors here, none of whom are listed, where they see this global epistasis in exactly the same data set I just showed you, the Ophelia Venturelli data set. So what I have is delta F on the y-axis and F on the x-axis, a plot for each species where that species is the species that's added to arbitrary background communities. That's the way this experiment is done. So now, what I actually have is for each species a slope and an intercept of this straight line. So that's n numbers instead of n squared. Right? It's 2n, say, right? Okay, even I can do that. It turns out that they see these patterns all over the place. This is another one of the data sets that we analyzed previously. Look at all those nice straight lines. It looks like this global epistasis is generic. Okay,
But a lot of them have slope close to zero, right?
I agree.
System with zero.
I agree. I went to visit Alvaro last year, and I was staring at these data with him, and I thought, I think I know an explanation for this. That's what I'm going to present to you. Okay, so there's all these straight lines. You will also notice what Daniel just said. Many of them are flat or very close to flat. Like, do you really take them the slopes very seriously? It's basically flat. And then there's just a handful that are truly crazy, pathological, relative to all of the other ones. Okay, so that's the thing to notice. But there seems to be a bit of an incongruence here because this only relies still, there's no dynamics, right? This whole picture only relies on presence absence. All I did was I measured the delta F when I added this species.
Just one, I mean, is this like delta F over F that you're plotting? Because otherwise the slope has no meaning, right? If you don't know what the units are. I mean, in the empirical data.
No, I think it's just delta. It's delta F and F.
So then there's no notion of large or small slope if you don't, compare the.
No, because delta F and F are the same unit.
Delta F and F are the same units, right?
Yeah.
I think
Well, but you
just
could say the natural is delta F times divided relative to F over a number of species or something like that. Because how big the perturbation is.
I don't know why the magnitude of the slope is important. I mean, it tells me the
Within a data set, it's not important.
what?
Within a data set, it's not important, but I guess across the examples you're
Okay.
I guess it's would I have expected this null would have been a slope of 1 over n or something like that. Because I've taken out one of n species.
Well, let's get to a null. I think I'm not a theorist, so I have to imagine it in reality, and it's going to turn out that not being in a Fourier basis is better. Okay, so there are straight lines and many flat lines. Yeah, and many more straight lines and flat lines. So, but what gives, right? Because now I have a characterization of the impact of adding every species to the community on the function, but I only need n numbers. I don't need n squared anymore. It's a lot less.
Sorry, so but when you say that there's not dynamics in your theory, but like, are you also in regimes in each of these cases where these have reached steady state?
That's the presumption, right? So in the Ophelia experiment that I published, and in this, I did not publish, in the Ophelia experiment that I analyzed, and the Ophelia and this experiment that we're analyzing, you can imagine the system goes to some point after the dynamics have played out, and you measure the function there.
Some have gone extinct at that point. Not.
I don't think so. No. This is like a one round of batch culture.
I see. Okay.
Yeah.
And they're in conditions where that actually happens. It's not just an imagine. Like,
Correct. Like there's something limiting the system. So the question is, what is the structure of these coefficients that gives me global epistasis? There's got to be some constraint, like, because when I fit these quadratic models, what I was saying was, okay, I've got, n squared of these coefficients and n of these coefficients, but I did global epistasis and I only need n numbers. Okay, so actually there's a preprint that from Alvaro last week, which I haven't really read carefully, to be honest. I'm very sorry, but I think it has even better support for what I'm about to say than I have. So I switched bases here to what I would call a Taylor basis, 0, 1, just because the algebra it becomes very, very simple in this Taylor basis. Okay, I'm going to make a guess, which you guys call an ansatz, very fancy word, which is that the quadratic effects of any given species I on J is just proportional with some constant that depends on J to the additive effect of species I. This makes the quadratic coefficients totally dependent on the first order coefficients, right? And collapses the number of numbers to n.
So this is like interaction via one chemical.
You're smarter than me, which I think we all already knew, but that's what we're gonna say. Yeah. But I think there's a bigger sociological message which I want to get to at the end. Okay, so if I compute delta F, I can just do this algebra. If I plug some things in, I get delta F is some constant, which I'm not going to write down for you, plus this Cj coefficient, which is specific to species J, times F of the community without J added. This is the global epistasis equation. Delta F equals constant plus slope times F. Same, no different. Okay. This is where I was at about 2 a.m. Now, if you go to one of these data sets where there's not too much noise and you can infer all of these coefficients, I can actually test whether this ansatz is true or not. 2:30 a.m. now. So it looks not bad. I was pretty excited.
What is plotted?
Sorry, what's plotted is the additive effect of the species that's added with its measured quadratic effect. And the lines are saying, oh, well, this one's pretty terrible, but the other ones, it looks like close to a straight line. And this, if you'll notice, this Cj, which is the slope of these lines, is my prediction for the global epistasis slope. And those two match. Okay? So what I'm saying is the global epistasis landscape is actually not that complicated, right? It just has n numbers. And those n numbers are cascading through the orders of the interactions in this statistical model. These things are all super correlated. And this is, I think, Alvaro's point as well. But I haven't, I want to extend this statement a little bit.
Do you do this for small s? Does it break down?
I don't know.
But why?
This is
It would.
this is 6 total. The max here is 6. I mean, if I have
6 is
two-
pretty high based on what Sarah did too. I mean, 6 is infinity for us, right?
That's true. That's true.
You just said that there was about all interacting via the total.
No, it was 6.
Okay. All right. Let's do this in a straightforward way. I'm going to write down a consumer resource model for this experiment, this butyrate production experiment. That consumer resource model has, this is what Terry Hua would write down if you asked him to do it. This consumer resource model has a biomass dynamics. It has that biomass is growing on some carbon resource, and then some coefficient beta determines how much of this byproduct that biomass produces when it's growing on that resource. And I'm sorry, the y's are all 1. Should have just. Yeah. So that means the picture is, if I have a community of 3 species, they're growing on some carbon, they produce some F, which is determined by how much of that carbon each one of those 3 species gets, and their beta coefficient, which determines their production of the byproduct on that carbon. That's my picture. Does it make sense? Okay. Now, here's the intuition that I think maybe Daniel already had. Imagine I have a 3 species community, and they're each getting a third of the pie of the initial resource, and they have some function. And now I amend this green species, which should be orange, but he's green. And that species consumes this fraction of the initial carbon resource, which is what this pie chart is, alpha j. That's a fraction, unitless number. Okay. Well, I can actually compute delta F if I know that alpha j. This is the byproduct that was gained. It turns out the fraction of the resource that was consumed by species j, which is alpha j, however much initial resource there was, times this conversion constant, which tells me how much byproduct that species produces when it consumes the input resource. And it turns out that the amount of resource that was lost is that alpha j C0, which was taken away from the fraction of resources, the resources that were consumed by the background community, times the average production of the byproduct by that background community. And the assumption here, which is a very strong one, which we'll break in a moment, is that the proportion of the resource used by the background community is preserved after I perturb it with species j. Okay, so if I take away this green wedge, all 3 species still get a third of what's left, for example. Under that assumption, this is true. And then I get this, which is the same thing. Again. Okay, so I have some intercept and some slope, which is now alpha j, which is the fraction of the resource that the added species consumes when you put it in. And I can do numerics, and the numerics basically show me, so here I make delta F against F plots for all of these perturbations, right, where I'm adding 1 species to all the backgrounds. And, if you look at the data, and you're never going to measure all of these points in an actual experiment, you would get an approximate straight line, I think, in both cases. And the analytical prediction is this dashed line, and the dotted line is the regression. And what's happening here is that when you add a species to a high-functioning community that grows very fast relative to the rest of the community, it distorts that resource pool across the other strains very strongly and causes this deviation from my analytical prediction. So I understand that deviation, I think, reasonably well. So this whole global epistasis picture comes from this alpha j, really, which is I added this species, it took some of the resources, and it caused this pattern that we saw. Fine. The statement I want to make is that alpha J just depends on the traits, the growth rates. I'm going to skip that. Okay. So what I did was I fit these complicated landscapes to this presence absence, and I predicted function from it, right? And then I thought, oh, I only need n squared numbers. And then I saw a global epistasis pattern, and I thought, oh, I only need n numbers. And now if I look at it from a mechanistic point of view, it turns out I only need n numbers, which are the growth rates or the yields or whatever. So if I had taken a mechanistic point of view of the function in the first place, I think I would have had a simpler description of the system. That is my argument.
It does say that the only thing that matters for the growth rates is the carbon.
It does say that. And if that's violated, you will see crazy stuff like that one species that has two vertical lines. But in the data that I showed you, almost everything is flat or negative, which is explained by this phenomenon. So my view is that this is a null. Under a competitive situation, you should always see global epistasis, and it's trivial in a certain sense. My postdoc Karnagauda, if he were here, he would have said, I told you this in 2022. And so if I have 5 minutes, can I present that, or what do I have? Okay. The statement that Karna was able to make a long time ago in my group was, all you need is the traits. And what that means is that if I understand, if I have measurements for how these organisms are growing and how they're using the resources, I can predict all of these things from that. Under the assumption that what Daniel said is not happening, that there's not some other crazy interaction going on in there, which is ancillary to the resources that I'm looking at that are defining the function of the system. So we've been studying this process for a long time. I'm not going to talk about it in any detail, but basically these organisms eat these molecules. Well, they don't actually eat them, but they use them to generate energy. They form this cascade of reactions which lets them generate ATP. That's all you need to know, called denitrification. And what we can do is we can make quantitative measurements of this in the lab, right? So I have organisms that do different subsets of these reaction steps. And I can go into the lab and I can make quantitative measurements of their utilization and production of these substrates. Like really painstaking experiments here. These data points are hard fought. I can take these dynamics, now the dynamics are here, and I can fit a consumer resource model. Why not? So Karna fits a consumer resource model. It now has a slightly different form because these resources are substitutable. I can take one for the other way around. I can fit that model to these data, and at the end, I get a phenotype, which is just these traits, order n numbers for a community of n species. And then I can say, okay, if I have two species and I have models that describe their metabolism, can I predict the dynamics of the metabolites in a community of two species with zero free parameters, just by integrating those models numerically, specified on each species alone? And the answer is yes. So the statement is that in this particular example, at least, if I know the traits of the individual organisms, I'm good in terms of now predicting the full dynamics, actually. It turns out that I can measure these traits for all of these species. This slide's out of order. And if I look at interactions across many pairs of species, the picture basically continues to work. That is, that if I have information about the traits of individual organisms, I can predict the dynamics of metabolites in communities. And it works, I'm going to skip this, up to 5 species in Karna's experiment. So my statement is this. We, meaning myself included, did a bunch of experiments to characterize these landscapes. I think under the psychological assumption that there was really complicated interactions going on in these systems. And then we found through simple characterization of those landscapes that actually they're quite simple, that there's only n numbers involved. And what I'm saying is, if I measure the traits of those organisms, that's n numbers. I can also make the same prediction of the community function as a function of the composition of the community. But these numbers mean something biologically. And therefore, my preference is for the right-hand side. That's the argument. Okay, I'm done. Karna did the experiments. Abby did the theory. Yeah, thank you.
Hey, Karthik from Yale with Purushottam Dixit. Yeah, we think about microbiomes as low-dimensional systems too, so this is great. But having said that, these experiments and the model seem to be for like single-step batch culture. Is that correct? Instead of like serial dilutions, which is what people typically do. So then, is it reasonable to assume that after the perturbation with like a single bacteria, the system has not really reached the steady state for that environment, and that it's still in like the linear regime, which is why you expect these the number of parameters to be like n, and if you did do serial dilution experiments, then like the biomass of each bacteria will change from step to step, and so the amount of resources that they consume will change and so on. So do you expect these results to hold up with multiple steps of serial dilution as well?
So Alvaro's last paper, I believe, did that, and the answer is yes. But I would certainly expect this characterization of the system, which is essentially dynamically complete, to hold up.
So I guess the next complication, since you mentioned nitrate and so on, if I say that there are two things that matter, so the carbon and the nitrate, and then but have the same one passage through. So you, instead of 2n numbers, you've got 4n or something like that.
Yeah.
Do you have a sense of whether that works, or is that something which is doable?
I think what happens is that essentially the ansatz that I made makes this interaction matrix rank 1.
Right, and then
which is n numbers.
Right, this makes it rank 2, but it's not quite an interaction matrix because the nonlinearity is associated with the growth.
I see. Yeah, you're right.
Talking about at lunch.
You're right. You're right.
Much more interesting,
right?
Because of the nonlinearities induced by the growth process.
So, is your question whether that interaction matrix then becomes n squared again?
No, it probably doesn't become n squared, but it's the structure. The n structure is sort of hidden in a nonlinear way.
Yeah.
Right. So,
I don't have evidence.
Might be able to do much
more. Could be trade-offs between the traits, right?
There could be trade-offs between the traits.
I think then the landscape would become more complex.
But even without trade-offs,
you just take any function that looks.
I probably should be able to answer this question by myself, but I can't. I guess what I've learned from working with you on these things is that the good variables that are supposed to enter these models depend on function. If you do it in an agnostic way, how does that change the conclusion about the simplicity that you're realizing at all, if at all? Does it matter, or for each function you can still do this? So,
I think for each function you can still do this. I think the reason that one would prefer the traits is because these numbers are now characterizing the mechanisms by which the organisms are proliferating. And so it's not really, there's actually, the good variables are the traits, but it's really the action of the traits on the resources that makes this so simple, right? The resources is the shared field that everybody's interacting with. And so the function that you're interested in should be biased, in my view, towards those functions that have to do with the biomass sustaining itself in the system in the first place, with growth and energy production and so on. I'm not sure that's an answer to your question, but we can talk.
So Seppe, I think some of the models that we show, for example, the calculation from the consumer resource model, you could see that the function depends on the initial condition, for example, of carbon, right? You had your C0. So presumably the dynamics also need the information of the initial condition in order to determine the outcome, which would be the function.
Sure. So n plus 1, you mean?
A what?
N plus 1?
I have no idea what that means. Oh, one parameter. No.
Yeah.
I rather mean that when you're analyzing the experiments, do you sort of assume that the experiment is some annealed average over all sorts of initial conditions that you might observe? So you couldn't.
No. So in the last set of experiments that I showed you, we provide the initial conditions.
So you know what they are?
Yes.
So it's initial condition dependent.
Absolutely. The dynamics will be initial condition dependent, right? On the biomass, for example. The time it takes to consume the resource will depend on the initial biomass.
Okay.
Yeah.
But in your microcosm experiments, you can conglomerate C naught with your what you call Ks, right? Like, sorry, like in the in your soil slurries, like you infer the combination. So.
Yeah, I think so. Okay.
Questions? See any? So we take what?
Can Theo ask one question?
Thanks for a really clear talk. I think I'm just a little confused about like the model's so simple it would never predict coexistence of these guys if you iterate, but then you do see the coexistence in the Alvaro style experiments. So I'm wondering if you have thoughts about that, or maybe a way these things are working is that they're learning some effective description after lots of complicated interactions have taken.
Where's the experiment where I see coexistence?
Just all of Alvaro's experiments, like the emergent coexistence stuff.
I think the question is ill-posed because in these experiments where we're talking about function, the question is for a single cycle, as I predict, as I measure, the function as a function of composition of the community, I can make this prediction. That's a one-shot experiment, right? So I think the coexistence question is a separate one. And actually what's happening in the experiments where Alvaro sees that long-term coexistence is the environment is actually much more complicated. There's spatial structure, there's temporal dependence on metabolism that's running and so on. So this consumer resource model is not valid, I don't think. But I think what's happening is the ecology is actually much more complicated in those systems.